let's get started talking about analytical methods and technology transfer. Specifically, how can we design and develop analytical methods with technology transfer in mind? Today, our goal is to try to answer that question at the following levels. Whether you're going from early proof of concept and moving into preclinical or a startup environment, or whether you're going through clinical phases from preclinic to the phase 1, phase 2, et cetera. And when it comes to managing your manufacturing partners, as well as from late stage clinical to commercial and beyond. So in the first part of our conversation, I want to talk about analytical method development strategy. So for whoever wants to start, please give us your thoughts on the importance of being proactive when thinking about the future needs as an asset moves between phases. And Cheryl, I see you on my screen first, so why don't we start with you and get your thoughts on there?
Okay, so I come in kind of midway, so I do the translational is what my team does. We have a facility that has TIs that bring their thoughts to us. We transition them into something that can go into a phase 1 or phase 2, and then hand that off to go into a phase 3 or commercial. So what we need to do is bring the analytical test from the PI into something that we can use for a release test for a product going to a human. So in these cases, we need to be able to validate it. So the important thing is to be using things that can be made robust, consistent. You need to use, when possible, you need to think about using reagents that are commercially available, because you're going to take these and you're going to give these out to other CMOs, whoever's going to be making your product. They need to be able to get the exact same things, reagents that are being consistent and be able to be used. When you think about your test, you need to look at what you're going to want to do for phase 1, phase 2, and phase 3. Phase 1 and phase 2 are generally going to be safety tests. And then, but you are also going to have a potency test as you go into that phase 2, phase 3 part, and of course into commercial. So in this, usually what we try to do is get a broad range of characterization tests and don't define a potency test for phase 1 and 2. We have all these tests that we actually back to the research, and they kind of do the research test. And then as we find what's important in it, then they can gradually narrow that down as they move through the different phases.
Thank you, Cheryl. Bill, Stefan, either of you want to add to that point?
Stefan?
Sure, yeah. Yeah, just to give you some background, so we have 2 commercial products already, and we have a pipeline. In terms of what to consider for method development and then going to transfer, it's important to understand have some line of sight thinking, right? It's important to establish a throughput, right, of the assay that is actually being able to just be reproduced not only, but also to be used frequently and in a QC environment, right? So this means that, and also with respect to the new ICH Q14 and Q2R2, there's some optional for now, right? So I work closely with the agencies. There's actually a standard coming out for method transfer, by the way, that went out for public consultation. That I have been leading for PDA, and there will be another round of publication consultation in next month. So I strongly recommend to take that as this is the latest and greatest that we as an industry and agencies think this is how it should be done. So, but important to understand that with Q14 out, and for the desire to actually use an analytical target profile, right, that is a living document that should be changed and will tell you the performance criteria that this test method should have initially, and then how it's staged towards commercial as well as post-commercials. And to start with analytical target profile, and choose your platform technology wisely, right? And that means has to be an instrumentation that is not only available globally, right, if you're expanding into global supply and licensing. You also need other things done as robustness studies are typically are actually now required to be done during development, and it was unclear before. So there's a change, right? It's no longer a validation activity, and it has to be completed prior to validation. That is a clear change in regulations or based in industry guidance, right? But essentially, this is what globally is now expected. And also the way robustness studies are done, right? You have to design those well, and then actually act on those where you do see significant trending when you're testing for your test procedural ranges, right? And another important thing, especially in the ATMP space, is the way you define criticality for reagents used, for assay control standards, and how these are strategically kind of coming on and get changed through the product life cycle. And it's particularly important through method transfer. You also should think early about how you do inventory management, right? As once you get to close to the commercial stage and in commercial, because you'll be manufacturing so much more and you'll be expanding globally so quickly, you quickly run out of everything. And so you have to do some planning and inventory management of your critical materials, starting materials, and the way you are managing technology changes, right? Through basically updating, you're always depending on the vendors that makes the instrumentation, their upgrades, their discontinuing of modeling of models, right? And we know very well that any kind of instrument change typically is a major a change for us, and typically requires not just a filing, but as the test results typically drift, also potentially a specification change and a major pre-approval submissions throughout the global stages, wherever you are in which country. It's also very important to have procedures that are sound, right, and that have a prescription in the actual execution part that is reproducible, it's understood and concise and comprehensive, so that you don't leave room for variation. In our space, we do a lot of manual processes still, more so than in traditional biologics now, as we are behind, right? Especially for things like potency testing, other assays, a lot of manual still. So the analyst performance is always a critical contributor to control and variation. And so it's very important to also do really, really good training management through development and transfers that are going to occur. And it's also important to stage your test system suitability testing requirements, so that you can be focused on throughput initially, and then you tighten those up as you move into commercial eventually, and through licensing. Also, what's important is that if you make autologous products, cell therapies, then the ramping up that you will have to do, this is also called capacity validation in the QC labs. That also has to be done staged, and your assay actually, from a configuration perspective, has to be capable on greatly expanding to get more samples on test, right? Then you have to think about configuration validation extensions. So the more you can kind of think about this up front, the more you would have to do add-ons, and the more issues you're going to avoid later when you actually have to quickly expand and simply test more and more frequently the samples that are going to come into QC. I think I'm going to stop there. I mean, I can tell you a lot more things what my experience has been, but I'm going to stop here and let Bill come next year.
Yeah. Thank you, Stefan. And Bill, I actually wanted to move into the second part of this conversation and get your thoughts on that, which is phase appropriate considerations for analytical development. I want to hear your thoughts on how you think the unique nature of advanced therapies influences development strategy, or should. And if you can please explain the differences between early stage analytics and later stage analytics.
Let me start with that last one. I think the big difference is not so much the analytics themselves, but the level of qualification and ultimately validation of the methods themselves. What is acceptable in early phase studies generally becomes increasingly less so. And I actually like to add to that, just to back up and add my 2 cents worth to what Charles and Stefan said, which is a deficiency in your analytics, which you can let slide by in the preclinical and even phase 1 clinical, does not get easier to solve as you go through the phases. In fact, if anything, it becomes more difficult and more problematic. I have actually seen a near crash and burn at the FDA because of an analytic method that didn't have its wrinkles ironed out in early phase development. And the party involved showed up at the submitted their BLA with these deficiencies actually still in place. Yeah, can you say whoops?
Yeah, right.
So there was a book that was published about 20 years ago called 7 Habits of Highly Successful People. And one of the chapters in the book, I loved the title, is Begin with the End in Mind. And the more one can look down and see where you're trying to get to and plan your analytics around that, actually makes everything go better. 2 other thoughts that I don't think either Stefan or Cheryl mentioned, but one is quality attributes. And I think some of these, particularly things like specificity of an assay, really, the sooner you can nail down the specificity of the assay, the sooner you know that that's actually going to be a suitable assay to move forward with. You don't want to get to phase 2 and discover that because all of the test articles you used when you were developing this assay didn't have anything in the matrix that might possibly confound the results, and you discover your specificity is only 50%. You can't use that assay. And the other thing that frequently gets overlooked is sampling. There's a tendency to, let's see what's the smallest sample size that we can get away with, because everything we take out as a sample is actually something that's no longer part of the product. The problem with that is as your sample size gets too small, the sensitivity of your assay falls through the bottom. And so those are things that I think need to be looked at. Now, getting back to, so the strategy is for moving through the phases of development is, I think one is actually to develop insofar as possible, or to measure insofar as possible, the quality attributes of the assay as early in the game as possible. You know, the bare minimum, you need to know specificity, you need to know lower limit of detection, and you really should know the linear range of your assay, so you actually kind of know what you're working with. And because those are things that, if they're not well documented, are also going to mess up your tech transfer. There's nothing more aggravating than trying to transfer an analytic method only to discover that the receiver of the technology can't get the quality attributes. So that would be the first. And the other, which actually goes to something that Cheryl said about making sure that your reagents are commercially available because other people have to use them. I would not only reemphasize that, but also reemphasize that you need to have an idea what the matrix. What if you're, say, taking a sample from a newly collected apheresis product, there's a specific matrix. There's other stuff other than what you're wanting to measure in there. And your assay needs to take that into account. And definitely needs to be validated with that being the matrix of operation. The other is that as you develop analytics, particularly process control analytics, for intermediate products, the methods you use to get to those need to be consistent, or if you change them, you need to requalify, because at the end, you need to actually have validation of your analytics on the product and its matrix as you will be manufacturing it for commercial purposes.